Welcome to this series of videos on integrated pest management, looking at disease control in cereals. In this episode, we're going to look at fungicide resistance because we're losing effective fungicides both through their withdrawal from use through regulation and their loss of efficacy through fungicide resistance. Now, so far we've been able to maintain effective disease control because the crop protection industry has managed to create new chemistry, usually just in time. But we've seen in other examples such as cabbage stem flea beetle in oilseed rape and blackgrass control on heavy land, where we've run out of control options. And this has created really serious problems for control and indeed our ability to grow crops. So we're going to look at what in IPM we can do to reduce the development of fungicide resistance. And we're fortunate in the UK that we have the Fungicide Resistance Action Group, or FRAG UK, which includes independent researchers and industry and keeps up to date with research and ensures that the guidance on fungicide resistance is in line with the evidence. And FRAG guidance can be found on the AHDB website. However, there are other opinions out there which haven't necessarily kept up with the research evidence. So let's go through what we can do to manage resistance and separate the wheat from the chaff. In this video on integrated pest management, we're focusing on how to manage fungicide resistance. So we can see from this list here that new modes of action of broad spectrum fungicides are relatively rare. Typically, a new mode of action is developed every 10 or 20 years, and we can't predict when the next new one is going to come along. So we need to look after the ones we've got. And the loss of performance that we often see following the introduction of a new mode of action or a new active substance within a mode of action can vary. In some cases, as in resistance against the azole fungicide group, the loss of performance can be gradual. We can see here field trial data over about a 15-year period showing a gradual reduction. This is in the case of control of Septoria tritici, which is very prone to becoming resistant. In other cases, the loss of control can be quite sudden and dramatic. This is the case of the QOIs or strobilurine fungicides. They were introduced in around 2001, and by 2004 there'd been a dramatic reduction in the level of field control that we could obtain. So there are various things that we can do to slow up that process and make modes of action last longer. And this video is going to be in two parts because it's a big topic. This video will deal with the effects of the number of fungicide applications, the dose we apply per application, the use of mixtures of fungicide modes of action, and the role of integrated pest management. The next video will deal with the topics of alternating modes of action, the question of whether it's better to alternate or to mix, and the effect of spray timing on fungicide resistance. But before we can answer this question, it's useful to understand the mechanisms by which fungicide resistance happens, because understanding those mechanisms will help us understand how to manage the problem. So there are 4 slides to follow that describe the mechanism, and then we'll get back to these practical questions. So first of all, if we think about how fungicides work, so all living things depend on a whole series of complex biochemical reactions in order to be able to function, and pathogenic fungi are no different in that regard. And those biochemical reactions are mediated by enzymes which act as catalysts to speed up the reaction. So in the top line, the enzyme is represented by the blue circle, and the chemical that's being affected by the biochemical pathway is represented by the red arrow. In this case, the substrate in red binds into the enzyme, into a particular binding pocket. The reaction is catalyzed, and that produces products of the reaction that often go on to another enzyme to be processed further. And fungicides are designed to block specific enzymes in order to stop the fungus being able to undertake those biochemical processes, and therefore block the growth of the fungus. So in the bottom row, we can see that a fungicide is designed to bind with the enzyme pocket more tightly than the natural substrate, and therefore prevent the natural substrate of the reaction from binding. Now, in the case of the vast majority of fungicides, they're targeting just one enzyme. They're termed single site acting fungicides. In a few cases, we have some fungicides which act on multiple enzymes, and they're termed multi-site acting fungicides. And you'll appreciate immediately, therefore, if a fungicide is only binding into one site in one enzyme, and the pathogen can change the shape of that enzyme in such a way that that particular fungicide can no longer bind, then it's quite simple for the pathogen to overcome the fungicide. Whereas if the fungicide's attacking multiple enzymes, it's much more difficult for a pathogen to overcome a multi-site fungicide. So here an enzyme is represented by a simple blue blob. Of course, enzymes are a bit more complicated than that. So this is an image of the appearance of an enzyme which is the target site CYP51 for azole fungicides, and all enzymes are proteins, and proteins are complicated molecules. They have complicated shapes, and if we look in the right hand diagram, we can see the little stick diagram of an azole molecule that's been designed to fit into and block the binding pocket of this particular enzyme. Now the protein is all sort of curled up to form its shape, but if we got hold of either end of this molecule and pulled it out straight, what we'd see is that it was made up of hundreds of amino acids, and those amino acids are in a particular order along the enzyme protein. Now the effect of that is that the shape of the binding pocket in the enzyme is dependent on that particular order of amino acids along the protein chain. And the implication of that is if one of those amino acids changes, then the shape of the protein changes, and that has the potential to mean that the fungicide can no longer bind as well. And we can see here, for example, some enzyme amino acids which are close to the binding site, and which therefore potentially, if the amino acid changes, the binding of the fungicide might change. And you'll hear sort of bandied about codes for different resistant strains against a particular fungicide, such as, for example, S524T, which we can see at the top here. And all that means is that the particular amino acid at location 524 along the protein chain has changed, in this case from serine to threonine, and that's enough to change the shape of the molecule and affect the binding of the fungicide. And in the case of azole resistance, we have very complicated situations of multiple mutations in the gene that codes for this protein, causing multiple changes in the amino acids, causing multiple changes to the enzyme, and multiple differences in the sensitivity of the pathogen that carries these mutations to the fungicide. So it's all hideously complicated. But fortunately, we don't need to worry too much about the molecular complexity of this, because the actual resistance evolution is determined purely by the resistance phenotype, in other words, how resistant the particular strain is. So let's just have a look how that works in practice. So on the left-hand side, we have a resistant strain which has one or more mutations, and a sensitive strain which doesn't carry those mutations. We start off with the sensitive strain predominating and the resistant strain at a very low level, but in the presence of the fungicide, the resistant strain clearly has a competitive advantage, and the population of that strain is going to grow more quickly than that of the sensitive strain. And that causes what we see on the right-hand side, which is the fraction of the population which is resistant increasing over time until we reach a point where the fraction resistant is high enough that we start to lose control in the field. So what we're trying to do is to slow up the increase in the fraction resistant. Now, there are two important points here which affect the strategies that we use to do that. The first is that this is not to do with the size of the pathogen population. The growth rate is about the per capita growth rate. So this is the amount of pathogen strain there is today compared to the amount of pathogen strain there was yesterday. So in evolutionary terms, an increase in the pathogen population from affecting, say, 0.1% of your crop to 1% of your crop, in other words, a tenfold increase, has the same evolutionary effect as an increase from 1% to 10% or from 10% to 100%. So this is not about the size of the population. And also on the right-hand side, the fraction resistant, which is what ultimately stops fungicides working, is also not about the size of the pathogen population. It's about the fraction of that population which is resistant. And we'll come back to why that matters in some of the examples later. Now, what this leads us with is a limited range of things that we can actually do if we want to slow down selection. If we want to slow down selection, we have to reduce the difference in the per capita growth rates between the resistant and the sensitive strains, or reduce the time over which that difference occurs, which is the time that the pathogen is exposed to the fungicide. So all resistance management boils down to three strategies. We can either try to reduce the growth rates of both the resistant and the sensitive strain. That will reduce the difference between their growth rates and slow selection for resistance. We can try to reduce the growth rate of the resistant strain relative to that of the sensitive strain, or we can reduce the time that the pathogen is exposed to the fungicide. And all of the resistance management tactics exploit one or more of these underlying three strategies. So let's come back to some practical examples. So the first one is to think about the effect of the number of fungicide applications. If we look at the data on the left, this is a mean of 4 field trials where an SDHI-azole mixture was applied in the field in replicated experiments, and we measured the amount of the resistant mutant in the field as a percentage or fraction of the pathogen population. Now, we can only do these types of field trials when there's already resistance known and in the field. So what we need to look at in the diagram on the left is the amount of mutants in the field, the frequency of mutations in the untreated, which is actually a measure of how much there was to start with before we applied any treatments. And in this case, we were already up at around 35%. What we're interested in is by how much the resistance is selected for above that dotted line. What you can see is if we apply one application, we get a certain increase in mutant frequency. If we apply 2 applications, we get twice the increase. And the reason for that is if we apply twice, we're doubling the exposure time. So it's the reverse of strategy 3, and therefore we would predict that increasing applications would increase resistance selection. So that's one example in Septoria triticale with SDHIs. We also have looked across the global research literature across all pathogens and all modes of action where this type of experiment was done, and we found when we published this work about 10 years ago 6 examples of studies where this was done, and in all of those studies, regardless of the pathogen or mode of action, increasing the number of sprays increased the selection for resistance. Now we come to a more difficult topic, which is the effective dose per application. Now, we might think that if we apply more dose, we're going to kill more of the bad guys, and therefore that would be a good thing. But in fact, the reverse is true. If we look at the data on the left, we can see the effect of applying an increasing dose, in this case again of SDHI, on the frequency of resistant mutants. And we can see along the bottom as we apply more SDHI, both the resistance frequency goes up and the selection for more highly resistant mutants also starts to be more of an issue. And the reason for that, counterintuitively, is that if we apply a higher dose, we're going to have a bigger effect on the growth rate of the sensitive strain than we're going to have on the growth rate of the resistant strain. Therefore, the per capita growth rates are going to be more different with a higher dose, and that is going to increase the rate of selection. Looking across the global evidence, we found 19 published studies where this type of experiment or modelling was done, and we found in 16 out of those 19 cases that increasing dose increased selection. Now, the difficulty of all that, of course, is that we want to be able to apply enough fungicide to be able to get really effective control of the disease. So one way to achieve that while still minimizing selection is to add a mixture partner. If we look on the left-hand side here, we can see a hypothetical case where we apply a solo fungicide of mode of action A, and that drives a difference between the growth rates of the resistant and sensitive strain, and that drives an increase in the fraction resistant. If we then imagine we had a different mode of action available to us and we mixed that with fungicide A, now the strains that are resistant and sensitive to mode of action A should ideally be sensitive to mode of action B, because there won't be cross resistance between the modes of action because they're targeting different enzymes. Therefore, fungicide B will reduce the growth rate of both the strains resistant and sensitive to A, and we see that in the top right-hand diagram. Therefore, the difference in the growth rates gets smaller and the increase in the fraction resistance becomes slower, which we can see in the bottom diagram. And if we look at some experimental evidence for that, on the left-hand side we have data from a set of mean of 4 field trials, which actually used an SDHI plus azole mixture, and the contrast here between the green and the yellow bars is that the green bars were applied without a multi-site mixture partner and the yellow bars were applied with a multi-site mixture partner. And again, we're interested in the increase over the dotted line. We can see that applying more SDHI increased resistance, but the inclusion of a multi-site mixture partner in this case reduced the amount of selection that occurred. And the full data for all of this and a lot more experiments are in the project report we can see at the bottom. So adding a mixture partner reduces the growth rate of both the resistant and the sensitive strains. It's a good case of strategy 1. And if we look across the global literature for lots more experiments that have tested this out, we found 49 studies where a mixture partner was used and the effect on selection measured, and in 43 out of those 49 cases the addition of a mixture partner decreased selection. So it's a very successful and reliable strategy. Which then brings us to the question about what to mix. So mixtures of modes of action reduce selection for resistance, and they have the benefit that mixtures increase efficacy and also sometimes the spectrum of activity across diseases. The choice of mixture partners needs to balance between minimizing resistance and ensuring we have an effective mixture that's also cost effective. And the reduction in selection for resistance is related to the efficacy of the mixture partner. So if you have an ineffective mixture partner, it's also going to be ineffective at providing protection against resistance. So multi-site fungicides, where they're available, and they're becoming rare because of regulation, multi-site fungicides are low resistance risk, so they're good mixture partners. Single-site fungicides are usually at moderate or high risk. So using a multi-site in a mixture with a single-site fungicide helps to protect the single-site fungicide. Mixing 2 single-site fungicides also provides mutual protection against resistance, but it also creates selection for resistance against both modes of action. But despite that, it's still a good resistance management tactic. But if a cost-effective multi-site is available, it's a good tactic to use it at the maximum permitted dose, and then to use the amount of single-site fungicide or fungicides that are needed to achieve the level of disease control that you require. And we also, of course, in all of this need to follow what is on the fungicide label. In some cases, those labels enshrine these resistance management practices as a legal requirement for use of the product. So this is an example for an SDHI product. It limits the number of applications that can be applied, and it also defines that SDHI fungicide should always be used in mixtures, and that the mixture partner should provide satisfactory disease control and must be of a different mode of action. We can also think more widely about mixtures. So from an IPM perspective, if we use a variety which is partially resistant against the disease, then it's going to slow down the growth rates of the fungicide resistant and fungicide sensitive strains, and by slowing down their growth rates, it's going to reduce the difference in the growth rate and therefore in principle reduce the amount of selection. There's very little field proof for this hypothesis, but what there is absolute proof for is if we use resistant varieties or other IPM practices that reduce the amount of fungicide that we need to apply, then there's very strong evidence that that will reduce the evolution of resistance and fungicides will last longer. So to conclude, different fungicide modes of action target different enzymes in the pathogen. Mutations affecting the target site enzyme of a single site fungicide are usually the major cause of resistance in fungicides. In insecticides and herbicides, that's not always the case, and different management strategies may therefore be needed. The total dose of a mode of action used in a season is probably the predominant driver of resistance selection. Decreasing the number of applications or the dose per application will slow down the selection for resistance. So we should use the amount of fungicide needed for effective control, but no more, and IPM practices that reduce the total dose needed are therefore the basis underpinning resistance management. And if you'd like to read more on this topic, there's the next video and there's some further reading here.